Okay, I'd like to start the next session, which is entitled, let's see, what is it entitled? It's something about building interventions and trade-offs in those interventions. And the word microbiome is in the title somewhere as well. So my name's Jordan Petja. I'm the moderator for this session from Yale University. Our work in my lab is focused on environmental biotechnology. So I wanted to say a few words about this session. This is, I think, an exciting part of the committee's work and potentially an exciting part of the committee's report. So we're going to ask some traditional questions about traditional building interventions and see if there's some unforeseen trade-offs. We're also going to consider microbial interventions that are not well characterized. And so in some aspects, this sort of looks into the future of what building and building design could be if it includes a building microbiome. So to help us through this section and through this process, we have an epidemiologist speaking today, an engineer, an architect, and then an expert in sustainable health design. So let's start off with the epidemiologist, Dr. Mark Mendell from the California Department of Public Health.
Thank you, Jordan. Am I speaking into the mic clearly enough? Yes? Good. Okay. So I was invited here to speak about a topic that I didn't think I knew anything about, so I decided to change the topic somewhat, but it's still something I really don't know anything about, so I should say that this is mostly stuff I've learned in the last week or two, and it's still very rough, so I hope you'll bear with me. And I want to talk about what is it that we know about trying to change the indoor microbial environment in ways that benefit human health? Because that seems to be maybe the ultimate question that people might want to be thinking about here. So there's a lot of ways that indoor environments are known to affect human health. They include microbial as well as a bunch of other kinds of exposure and mechanisms. As an epidemiologist studying indoor air and health for years, I've always thought of the microbial influences as indoor microbial exposures. You know, exposures, adverse, that's the way we thought about it. Now we learn that there is something called indoor microbial communities, much more complicated. And then we learned that there is the microbiome, which is even more of a grand concept and complicated. And there's a fair amount of research now suggesting that there's relationships between microbial aspects of the indoor environment, not measured necessarily, but things like forms that you might think of as microbial, and human health. And there's some research on what does the indoor environment have to do with the indoor microbial environment, and then what does the indoor microbial environment have to do with human health. But we don't know a lot about any of these. And so this is the kind of map I'm going to try and walk through and see what we know about these connections. And the particularly confusing part about it is that the link between the indoor microbial community and health seems to have both good and bad effects, which seems confusing. So I'm going to very briefly review findings on a number of relationships. I'm going to kind of map this out here, and it's still rough, as I said, so there's a lot of material here. So it's going to look at environmental factors that are microbially related. For instance, do you see dampness or mold in buildings? It's not measured, it's just a factor in an environment, indoor environment, it's microbially related. There's now a pretty large literature on adverse health effects related to aspects of the environment like visible mold, mold odor, dampness and such, and adverse health effects. Then there's also research on what can you measure microbiologically in the indoor environment. Usually they're looking at specific taxa, we used to call them species, I guess, and compounds that are microbial and adverse health effects. And then people also looked at how does the environment influence which of these microbial taxa are present. And then more recently, we find that there are environmental factors that are microbially related that seem to have beneficial health effects. And then people have also looked at what are the measured microbial community factors, diversity, richness and stuff, and how beneficial health effects are associated with them. And then people have looked at what is it about these environments? What are the microbial community factors related to the environment? So there's a lot of different connections here, each of which has sometimes quite separate research studies. And so I'm going to try and run through what we know as of about a week or two ago about all these relationships. And so it's going to be messy. So there's microbially related environmental factors that are related to adverse human health effects. The most well understood is that observed dampness or mold are consistently linked with increased respiratory and allergic disease, and there's been some good research suggesting that, or demonstrating that when you remove dampness and mold in buildings, it decreases the health effects. There is three actually interesting studies suggesting that in the presence of observed dampness and mold in homes, lower ventilation rate increases the risk. Probably it's increasing the exposures coming from the sources, the dampness sources in the buildings. Then something which seemed relevant here is that there's disease transmitted from other occupants, which I am told would be considered part of the microbiome, the indoor microbiome. So from surfaces, you get disease agents that cause more respiratory and gastrointestinal infections, and it seems to be accepted practice that cleaning and disinfecting reduces this transmission. In aerosol, when you have long-range aerosol transmission of infectious disease, you get more respiratory infections indoors. And as I understand it, in healthcare settings, it seems to be well accepted that increased ventilation and air filtration reduces transmission, but only if it's long-range airborne transmission. And then I'll go into this more later, but if you think of non-farm environments as being environments that produce adverse health effects, you could talk about all that right here. I'll talk about it instead as the presence of farm environments as having beneficial effect. So there's a lot of evidence. I'm going to go into this particular bit in more detail because this is what I know about most. Observed indoor dampness and mold is very consistently and extensively linked to respiratory and allergic health effects. And these include factors like visible mold, mold odor, moisture or dampness inside, and water damage in buildings. And this is the conclusions from 3 studies going from less recent to more recent, showing what the strength of the evidence is. And the symbols in this little chart, as they go up and get darker, they're showing increasing strength of evidence. So there have been slowly increasing numbers of health effects. These diseases and these symptoms that have been consistently linked to observed indoor dampness and mold. And as of this last article, they actually concluded there was a causal link demonstrated between observed dampness and mold and asthma exacerbation. An example of a really nice index of visible mold and water damage and dampness comes from Tina Repinen's group in Cincinnati, where, without going into all the details, there's an index of 3 levels of observed dampness or mold, and you get a nice dose response for all these outcomes in 4 published studies at ages of 1 year and 3 years, with very high risks at the high level of odds ratios, which is essentially a multiple of the baseline risk of 3, 4, 5, 5, 4, 6, 7, 9, and in allergic kids, 42 for observed dampness and mold. So this is a very good evidence of a strong link there. And then we go to farm environments. So we have found that there's consistent benefits, reduced risk of asthma and allergy from growing up on a farm. Amazing. We don't understand it yet. We don't know the exposures. We don't know the mechanisms, either of the farm environments that are beneficial or the damp and moldy houses that are harmful. So building dampness or mold is associated with increases in these health effects. These are harmful exposures to something microbial in buildings. Farming environments or farming activities reduce the risk of allergy and asthma, so these are beneficial exposures. We don't understand this. So getting into what some of the specific research findings are, farm environments have beneficial health effects, and people have now done a bunch of studies trying to figure out what is it about farm environments? What are the actual mediators of this beneficial effect? So it turns out that these effects are particularly strong in utero. It turns out that the effect may even be stronger when the exposure is to pregnant women and the benefit for their kid when the kid is born. And these environments, again, in utero or in early childhood, reduce risk of later asthma, atopy, or hay fever. And these are the specifics that people have found that tend to explain the farming effect. And so it includes animal sheds, stables, cattle, pig keeping, and a dose response with a number of different animal species, say, that the pregnant mother encountered in her life. Hay, hay lofts, silage. In case you don't know what silage is, farmers rot hay so their cattle can eat it, and that's what a silo looked like. And there's a really interesting paradox here. When you study respiratory medicine, it turns out that there's a serious respiratory hypersensitivity disease called hypersensitivity pneumonitis, which can be disabling. One of the classic occupational causes of hypersensitivity pneumonitis is silo uncapper's disease. So the person who would open the silo after it had been rotting the hay all winter, they would get really sick. They would get respiratory disease. And yet, exposure to silage is protective. So just one of the many paradoxes here. Grain-associated activities. You have to drink the unpasteurized farm milk, and it's strongly protective against allergies and asthma. A large family, independent of farming, has these protective effects. They've done studies where they've teased those apart. They can take the dust from houses that are in farms and reproduce these protective effects in mice unless they alter the mice so they have a lack of a certain immune signaling molecule, and then that protective effect goes away. That's very interesting new research, as is this, where they've looked at low-dose endotoxin in the house dust. Oh, I'm sorry. The house dust has lots more endotoxin. So they also used just endotoxin and found it caused the same benefits in mice as the farm dust, unless they removed a certain enzyme from the mice, and it reduces specific lung cytokines that lead to sensitization, but when they get rid of this enzyme in the mouse, it doesn't happen anymore. So they actually seem to be focusing and honing in on maybe the exposures and the mechanisms behind the farm effect, which is really fascinating. Then non-farm environments also have some beneficial health effects. Turns out dog exposure in early infancy has been shown to be protective in a bunch of studies, and it turns out it alters the house dust. And when they use this house dust in mice, they get an altered gut microbiome, which has beneficial organisms in the gut microbiome in the mice. And it protects the mice against allergens, viral infection, and improves their immune parameters. It turns out that they've done sort of a series of studies across less and less developed countries, and they find that the less developed the country, the more matured the immune response in infants is. Again, this suggestion that greater development leads to environments that are somehow not healthy for us. And there's one really interesting study that looks at the question of cleanliness, because the hygiene hypothesis, which everyone knows about, many people think it's that we clean too much. But these people looked specifically at that issue and found that personal cleanliness, greater personal cleanliness, and greater home cleanliness did influence parameters in the dust. It did not influence risk of asthma or allergies at all. And so their conclusion was that microbial parameters are clearly related to risk of asthma or allergies, but microbial parameters that are not related to personal or home cleanliness. So this is on the relationship of the environmental factors to the to the microbial communities. So living on farms clearly is related to greater microbial diversity, mostly bacteria measured, but a few studies have looked at fungi. Pets or dogs indoors, greater bacterial diversity, and a greater abundance of dog-associated taxa. These factors have all been associated with greater fungal richness. Suburban versus urban homes, water leaks, wet walls, flooding, and more extensive use of air conditioning. And it turns out that, I think this is from the Oregon group, that higher ventilation rates reduce the relative abundance of human pathogen-like bacteria, providing some confirmation that ventilation, increasing ventilation may improve, may reduce transmission of indoor pathogens. People have also looked at measured indoor microbial taxa and the environmental factors, living on a farm, again, greater microbial diversity and higher endotoxin. There's certain specific taxa that show up at higher levels in dust in farms. Greater occupant density improves some beneficial microorganisms. And it turns out that everyone knows this apparently, that specific human microbiota have a really predominant, well, a strong influence on the occupied home microbiome. And then there's the question of, is anything that we know how to measure microbially shown to be related to adverse health effects? So low bacterial diversity, again, this is the flip of high bacterial diversity being protective. Low bacterial diversity you could see as harmful. High bacterial richness, this is in direct conflict with the first point, and it's one of the paradoxes we have here. As to specific, say, fungi or bacteria studied and found consistently linked to disease like asthma or allergies, none have yet been consistently linked to my knowledge. But there's a lot of suggestive findings. So hydrophilic fungi which like moisture, have been shown in two kinds of studies to be related to asthma. One is the NIOSH studies, which used culture, and the other is the Cincinnati studies, again, which used qPCR, and they used a metric of ERMI to group the hydrophilic fungi. Other studies have found, again, this surprised me, that very short-term culture samples in indoor air found that higher concentrations of culturable penicillium and of total fungi in air were pretty consistently related to asthma exacerbation, but just in a few studies so far. Another studies found that if you sum the allergenic fungal species and then specific other metrics, you get a relationship to greater asthma severity. The microbial chemicals of endotoxin and glucan have been associated in some studies with increased wheeze, but in other studies they're protective. And muramic acid in mice increased the immunotoxic potential. They come from gram-positive bacteria. Then, are there any measured indoor microbial factors, whether communities or taxa, that have links with beneficial health effects? And all the things I'm going to show you are associated with reductions in either asthma, allergy, or wheeze. So greater microbial diversity, a bunch of studies have found to be associated with beneficial health effects. Specific bacteria in various studies have been found associated with the beneficial health effects. So these are, I think, trying to find out what's behind the farm effect. Specific fungi, a few. And again, these microbial chemicals are associated with beneficial, but sometimes adverse effects. We don't really know what's going on. And another study found total microbial markers, but not the specific ones, was associated with beneficial health effects. And then the most interesting of all these to me is something I heard presented at a conference recently, but it has not yet been published, which is in a European study. They studied farm homes, non-farm homes, took a lot of samples and next-generation sequencing, and then used various statistical metrics to tease out what specific combination or signal in the microbial world, the combined bacterial signals, best explained farm versus non-farm. And they found that when they came up with a mixture, and when they put this in their statistical models, farm disappeared from the model. This completely explained the farm effect. So this is very exciting if it's actually going to hold up. And that's something we would really like to know. And in fact, they found that there were some farm homes that were not farm-like and some urban homes that were farm-like, and this particular metric kind of took care of that, and farm-likeness was the protective factor. So really what we know about the biology can't explain what we're seeing in the epidemiology. We know that some of these early microbial exposures reduce later atopy and asthma, but this is something I think is really important. Dampness or mold exposure at any age consistently increase respiratory risks, whether you're an infant or a child, an adult, or an adult. So it is not clear, as some people might think, that all early microbial exposures are good. Only some kinds are good, and we don't know exactly what they are yet. So this is paradoxical that different exposures can have these different effects. So to try and summarize this all very briefly, we know that indoor dampness or mold is associated with this increase in these health effects. We don't really know what the microbial agents are, but at this point it seems they might be hydrophilic fungi, maybe some of the microbial compounds. For disease agents on indoor surfaces, as I understand it, they cause increase in respiratory and gastrointestinal infections, and they are virus or bacteria. For indoor air disease agents, we know respiratory infectious agents that we believe are transmitted through long-range transmission in air, virus and bacteria. If you think of the lack of diverse microbial exposures as a harmful situation, then you could say that a lack of diverse microbial exposures increases asthma and allergy, and the only agents we know about are that they're less diverse. And I should make a little point about diversity of the microbiome. If farms are associated with protection against disease, farms are associated with greater microbial diversity, some people might conclude that greater microbial diversity per se is the beneficial factor. But I don't think we know that, and that would be premature to say that, because you can imagine that a more diverse microbiome in a home would be beneficial simply because it's more diverse and it helps educate the immune system. Or you could imagine that simply some particular rare taxa are able to be present in a diverse microbiome, and they're the positive influences, and the diversity itself is completely irrelevant, and we don't really know which is the case. So this cocktail of beneficial bacteria that they've identified in the European study may be completely separate from whether it's actually more diverse. I don't think we know that yet. And so a lack of a farm-like environment could be seen as a risk factor, and this is a lack of exposure to animals, lack of exposure to animal feeds, and again, we don't know what the specifics are there, but this table kind of summarizes overall what we know. And it'll be difficult if we want to try to make interventions to change the indoor microbiome to improve the health of occupants. There's a lot of challenges there. First of all, do we need to document actual causation before we go about changing indoor environments? Or how sure do we need to know about all this before we actually start changing buildings? If we're going to change the buildings through their design or the materials we use or how they're operated or how they're maintained, do we want to reduce the adverse exposures, say the dampness and mold we know about? Do we want to increase the beneficial exposures? Do we need to do both? It would be very difficult at this point to identify and then provide all the missing beneficial factors, because we don't understand them, in an intervention. And I think it would be best if we could create some kind of self-sustaining beneficial process which would have to be analogous with the fecal transplants in humans, because when you do that in humans, it takes care of itself and maintains itself. Not clear how we could do that in buildings, because you seem to need to live near a farm with farm materials coming in constantly. One of the European studies found that animals that go in and out was a good piece of the beneficial effect, and that people were, bringing in things from the barn was part of that process. So it's not clear how you would change the urban living environment to reproduce that kind of beneficial exposure. So based on all the knowledge that we have available, some ideas for what kind of interventions might make sense. For indoor dampness and mold, of course, there's getting rid of the moisture source, the wet materials, and the moldy materials. Increasing the outdoor air ventilation does reduce indoor humidity, and also it would reduce the exposures related to indoor dampness sources. For disease agents on indoor surfaces, it seems to be accepted practice that you clean and disinfect often touched surfaces, although one study found that the common commercial antibacterial products were useless. For disease agents in indoor air, I think it's accepted practice in healthcare settings that you use isolation, increased outdoor air ventilation, and increased particle filtration. But I think there's very limited evidence for doing any of those things in general buildings because it's not really clear what increased ventilation gives you in terms of reduced infectious disease transmission, although there's a bunch of studies that have suggested that increased ventilation reduces illness absence in offices and schools, which may be from infectious disease transmission. For lack of diverse microbial exposures, somehow we'd want to add a diverse microbial cocktail. We don't really know how to do that. And then for lack of a farm-like environment, I can come up with lots of interesting ideas. We could add some kind of microbial cocktail to buildings. We could try barn fecal transplants to homes. Because there's one noted expert I talked to, I asked him what he thought the farm effect was all about, and he used a four-letter word for excrement. He said, that's what it's all about, so maybe it is. We could do barn-to-home carpet rotation cycles. You could have indoor cattle or pigs as pets. You could have hay-based furniture. Or you could have, and I think that that the raw milk has to be drunk, but if it had a more general influence, you could have raw milk decorative fountains. And so Dick Hedrick and I proposed in the Sloan Coopio meeting, we were supposed to come up with practical, useful studies. So we came up with the split pig study, or no, it was the twin pig study, where you have townhouses with a common wall, and you comfortably embed a pig in the wall. And one of the families gets to experience the relationship with the pig, and the family that gets the business end of the pig gets to experience its gut microbiome, and you see which is actually, what is it that you're relating to the animals on the farms, or is it the microbial exposures? Anyway, that's it. So, thank you.
Thank you, Mark. Mark went a few minutes over time, but I think it was worth the few slides on the interventions. Our next speaker is Dr. Michael Waring from Drexel University, who's going to speak about green walls and houseplants.